Good morning, students. In this session I am going to discuss the physical property of amino acids. So physical property of amino acid, as we have discussed the more about the amino acid, essential, non-essential, they are optically active in amino acids, so on. So physical properties, the first, it is colorless, non-volatile, crystalline solid, insoluble in water, insoluble in non-polar solvents, in aqueous solution, amino acids behave like dipolar ion, which is called Zwitter ion. So let's discuss what is Zwitter ion. So at first we discuss about the structure of Zwitter ion. Suppose we have a compound like this is R, and one side this group present, one side this group present, and one side group this is present. So here is the positive charge appears on the nitrogen, means it has tendency to give the protons, and in the aqueous form it is also behave like NH3 plus, and here I am also representing one configuration minus, here is H and here is the NH2. So see here, it is the acidic form because it releases H plus ion. So we can write here, this is the acidic form because it has tendency to release H plus ion. And it is the basic form because here is a lone pair present, so it is called the basic form because it has tendency to donate electron pair, and this form is called the neutral form. Why? Because this has tendency to accept, because here is this group is called the basic group because it has tendency to accept proton, and this group is called the acidic form. It is called the acidic form because it has tendency to release H plus ion. Basic form and acidic form. So in this particular form, here is the negative charge present on the oxygen. So in that condition, this carboxylate ion quickly accepted proton, and this NH3 group has tendency to release H plus ion. So in this group, both basic group and acidic groups are present. So this is called the neutral form, and this neutral form is called the Zwitter ion. So once again I am describing, when in amino acid there is a carboxylic acid present and amine group present. In amine group there is a lone pair electron, so it has tendency to accept protons from the water, because in aqueous solution we are discussing about the aqueous solution. So it has tendency to accept proton from the water. Simultaneously this acid has tendency to give H plus ion because it's the acidic group. This has tendency to accept H plus ion because this is basic group. So in that condition we can say that amino acid behave like a dipolar ion in the aqueous solution. So let's define the definition of Zwitter ion. Simply write down the Zwitter ion. What is Zwitter ion? Zwitter ion. So Zwitter ion is an amphoteric. It is an amphoteric because behave like a acidic as well as basic. Amphoteric ion. Amphoteric ion, and during electrolysis it neither migrate to negative means cathode electrode nor anode. So we have discussed here as CH and NH2 if we provide H plus or OH minus. So CH, COO minus, and due to presence of lone pair is accept H plus from the water, or you can represent by this also. COH in presence of H plus or OH. In water, as we know, H plus ion and OH ion is present. So we can simply define in basic medium. Suppose this is the K1 form and this is the K2 form. In basic medium goes towards carboxyl ion or carboxyl ion. In acidic medium goes towards ammonium ion. So an ammoniated and amino acid never exists in uncharged form. Simply we can say that an amino acid in aqueous form, we are discussing about amino acid in aqueous form. Amino acid never exists in an uncharged form. So it is simply discussion about the Zwitter ion. Let's we discuss about the protein. Let's we discuss about the protein. Chemically, proteins are condensation polymer, actually polyamide, in which in which the monomeric units, the monomeric units are the alpha amino acids. Simply chemically we can distinguish. So an amino acid is, so next definition is the, you can write here also, an amino acid, we have already discussed about the an amino acid. As the name suggests, it contains amine group or which is called acetic group. So, or we can say that large proteins are large biological polymers of alpha amino acid linked by peptide bonds. Or we can say that proteins are large biological polymer of alpha amino acids linked by peptide bond. So on classification of proteins, at first we classified the proteins so classification of proteins. Number 1, on the basis of molecular structure. So it is the two type. First is the fibrous protein, and second one is globular protein. So fibrous protein, it consists linear thread-like molecules which tend to lie side by side to form fibers. Next property is it is insoluble in water. And third property is polypeptide chain in them are held together usually at many points by hydrogen bond or some disulfide bond. So fibrous protein, there is a presence of polypeptide chains in them are held together usually at many points by hydrogen bond and some points disulfide bond, disulfide bond, disulfide bond. So intermolecular force are strong. So we can say that intermolecular force are strong due to presence of H bond or polypeptide bond. So intermolecular force number 4, intermolecular force are strong. So let's we discuss the globular form of proteins. Globular form. Globular proteins. In this type of protein, in this type of proteins, the molecules are folded together into compact form, the peptide chain are held together by hydrogen bond. They are soluble in water. Example, enzymes, hormones, etc. They are spiral shape. Spiral shape. So let's discuss the peptide bond. What is peptide bond at first? Peptides are amides. Formed by the condensation of amino group and group of alpha amino acid with the carboxyl group of another molecule of the same or different alpha amino acid with the elimination of a molecule of water, elimination of the molecule of water. They are classified as a di, tri, tetra, petra according as a 2, 3 forms molecules of same or different amino acid combine together. For example, NH2, CH2, C double bond O and OH. This is the example of glycine. And another amino acid, suppose we take NH2, NH and NH2, CH, CH3 and COOH. So the removal of this OH and this H, so this is the structure of alanine. So by removing H2O, by the condensation of this two amino acid, so formation of compound NH2, CH2, C, O, NH, CH, CH3 and COOH. This CONH group is called peptide bond. Peptide bond. So the CONH group bond peptide bond or peptide linkage. We can say that the CONH group is called, this CONH group is called the peptide bond or peptide linkage. So let's we discuss the denaturation of proteins. So denaturation of proteins. It is also an important topic. Denaturation of protein is an important topic. A process that changes, the physical and biological properties of protein, physical and biological properties of protein without changing the chemical composition. Protein without changing the chemical composition of proteins is called denaturation of protein. It is caused by the changes in pH, temperature, pressure, presence of salt, etc. Important features of denaturation. Important features of denaturation. Of protein. It does not change the primary structure but changes the secondary structure of protein. For example, boiling of a most common example. So let's we discuss the type of proteins, primary, secondary, tertiary. So let's we discuss the type of proteins. Structure of proteins. Structure of proteins. So at first write down the structure of proteins. First it is primary structure, primary, and secondary structure. Secondary structure further divided into two parts: alpha helix structure and beta plated sheet structure. At first we talk about the primary structure. So write down the primary structure. It tells the sequence in which various alpha amino acid, alpha amino acids are present in the polypeptide chain. Polypeptide chain. Each protein has a specific sequence of alpha amino acids. If we change the sequence of any alpha amino acids, then the biological functions of protein get changed. Then the biological functions of protein get changed. So suppose this type of change, suppose this is this type of changes arrangement like this, or we can say that if we have red beads and yellow beads and blue beads, suppose. So arrangement like this, suppose red, blue, yellow, red, blue, yellow, red, blue, yellow. So a specific sequence it has. If we change the sequence of any one, so their biological functions get changed. So this is the structure about the primary proteins. Let's we discuss the secondary structure, in which we talk about the alpha helix structure or beta helix structure. So students, in the next video we will discuss these two structure, and also about the nucleic acid, in which we will discuss about the DNA and RNA. That's enough for today.